So with that, would like to introduce our first speaker. Dr. Karen Anderson is one of my colleagues in the Division of Hematology and Medical Oncology. She's a world-class expert in breast cancer, a world-class immunologist who also is at Arizona State University, where she does really pioneering work regarding the diagnosis of cancer as well as new cancer treatments that are based on the immune system. She even teaches the students at Arizona State University about the immune system in people that have trajectories that are going to be researchers or medical students or scientists. So I could think of no one better to really try to share with us how we are trying to harness your immune system to fight cancer. Thank you.
Thank you so much. This is such an honor to be here. I talked here a couple years ago, and as I was talking with Dr. Mesa, it really has been a transformative change in the field, even within the last couple of years. And so what I'd like to do is update you some about how cancer immunology has progressed in the last several years, how it is now impacting our patients really on a routine basis now, and maybe give a little bit of a glimpse as to where it's going to come in the future. One of my caveats is that I do teach immunology at ASU, and so I have graphs and charts and structures and things, but I think that it is important to, when you're starting to sift through all the clinical trials and the media, and to try to understand how these therapies are coming about, to understand what they are and how they might impact your care. This is back from 2013, so we're now 4 years later. In the scientific journals, it was heralded as the breakthroughs of the year for cancer immunotherapy. It has long since hit the mainstream media, and have really emphasized, and we've started to integrate immunotherapy in terms of our routine practice. But this is not a new thing. Cancer immunotherapy has been around for over 100 years. The first real evidence that we knew of for efforts at cancer immunotherapy was something called Coley's toxins, which were essentially streptococcus. It was a bacterial infection that was sort of ground up and used as an injection into a sarcoma. This was in New York City back in the late 1800s. And what they found was that a few patients seemed to respond to this, and they couldn't really understand why. And so the problem was we really didn't understand how the immune system was working until you got back into the lab and you started to understand the individual little molecules that were involved in activating and impairing the immune system. And part of the reason is that our immune system is a balance. It is this inflammation that can cause cancer. It is this anti-tumor immunity that keeps it under control. Your cancer has been growing up and evolving, if it's a solid tumor, probably over decades in the presence of your immune system, so it has evolved ways to evade it, or it wouldn't have survived. In fact, probably we have many cells that are continually being eliminated by our immune system as they mutate and change long before they ever develop cancer. And so to really understand how you manipulate it, it has to be a very focused effect on the cells that affect cancer versus the ones that cause inflammation or actually cancer promoting. And so in some of the major highlights that have really changed our understanding of cancer, one of them was the identification of what we call checkpoint blockade, the molecules that keep the immune system in check, which really started in the late 1990s by Jim Allison. And then cytokines, which are the messengers of the immune system, started being used more routinely, in particular for the treatment of melanoma, where in renal cell cancer, where a lot of this was pioneered, and then now more generally in others. We now have targeted therapies for the opposite. Rather than trying to activate certain parts of the immune system, the autoimmune field has identified molecules that where the immune system is gotten too active, and so they have a series of targeted inhibitors, obviously, to try to turn down the immune system. And so it's really this balance between how does one target the cancer without driving too much autoimmunity, which has been one of the real challenges within the field. So, let's see if I can do this. So I think the questions that we hear from you on a regular basis in clinic is, what is inflammation? Is this a good thing? Is this a bad thing? How do I drive an immune reaction to my cancer? And then what type of immunotherapies are out there? I'm hearing about all of these. How do I find out which ones are available for me, which ones would make sense for my cancer, my tumor type, and what's coming down the pike? So first I want to tackle a little bit about inflammation. All right, what we know is that a host of things can cause inflammation. I would say there's more out there in some ways on inflammation in the lay literature than there is almost in the scientific literature. It's something that we all talk about, but we're not exactly sure sometimes what it means and how to target it. So what we know is that microbes and our microbiome in particular can alter our general, inflammatory processes. What we know is that the immune system learns a tremendous amount very early on from gut organisms. And as an immunologist, and we have millions and millions of organisms that we are co-hosting, and one might ask who's driving who sometimes. We're perhaps merely the host for all of our happy little bacteria, and we want to keep them happy, and they'll keep us well. But these microbial pathogens, and understanding how do they actually cause chronic inflammation, and how do our diets alter that, I think really is an area of very active investigation. Mayo Clinic and Arizona State University have a number of different projects actually looking at those very specific questions. But one of those challenges, it's not a simple thing, obviously, and I think we have a lot still to learn about how does one alter diet, how does one change your microbial environment, all right, to alter inflammation, in particular within the gut, where a lot of this focus has been, to change either risk of cancer or risk of developing a cancer recurrence. And I think that this is an area that is really under active investigation. It's still in development. We know that chronic inflammation contributes to cell transformation, to growth, to metastases for a number of different cancers. But one of the things that we know is that integrity of the gut lining is altered by the composition of the microbes that are there. And those pathogenic conditions are the ones that cause disease, all right, can cause essentially a break within the gut lining and that inflammation, and then that causes a recruitment of immune cells. And those immune cells then cause somewhat more chronic inflammation. And we have these regulatory T cells that kind of keep it under check. And altering the composition of these regulatory T cells, which basically turn off that inflammatory signals, and we can count these and we can measure these and we can have animal models that look at these, and finding that right balance between active, reactions against gut microbes to chronic inflammation that contributes to disease is one of the challenges. So we actually have at our disposal an incredible therapy, okay, an incredible anti-inflammatory therapy that has been around for 2,000 years. And so one of the questions has been, well, what role does something as simple as aspirin or anti-inflammatories, which many of us use on a regular basis, what role does that have in cancer development? What role does that have, should we be thinking about this? And so it's not a simple question, because things like anti-inflammatories, as we know, and aspirin in particular, if you look at the effects of low-dose aspirin, over time, okay, can reduce the risk of cancers, okay, in a number of different studies, okay, if there's chronic use, but it is also associated with increased risk of bleeding. So many of us, there's been many studies about the impact of low-dose aspirin on cardiovascular disease. All right, and that's recommended in certain populations for prevention of cardiovascular disease. But the real question, and we're trying to figure this out, should we be thinking about aspirin to reduce recurrence rates? Okay, simple anti-inflammatory, should we be thinking about this for recurrence rates, and in particular for breast and colon cancers? And so what we know is that there's been a meta-analysis of 8 different randomized trials involving thousands of patients looking at the role of aspirin. And what they found is that there is a mild impact on reducing the risk of cancer death with the use of aspirin, in particular for colorectal cancer, but also impacting other cancers. What those studies have led to is there's currently the ESPRIT trial, which involves 19,000 healthy people, all right, people over the age of 70. This is a not just multinational study, okay, it's involving Australia and the US and a number of others at many sites, to ask the simple question, does aspirin help prevent cancers and other diseases? They're looking at a host of other diseases in healthy people over the age of 70. We're hoping to have information and results from that. The trial's actually closed. We're looking to get the data from that by next year. So hopefully we'll have some better guidance for people. Should we be thinking about aspirin or not? It is not a simple question to say we need to take this because of the bleeding risk. So we need to be informed on this. But there are 2 additional trials that are ongoing, and I am thrilled that they are. One is called the ASCOLT trial, which is that involves 1,200 people with colorectal cancer to ask, does aspirin help prevent recurrence? And so that trial is ongoing, and it's ongoing because we don't know which is better, all right? And then there's also the ABC trial, which recently opened. I am passionately in favor of this. This involves 3,000 women nationally with breast cancer. To ask, does aspirin help prevent breast cancer recurrence? And the prediction is it might within subsets of breast cancers, but we don't know which ones. This was based on an observation from 10 years ago, all right, that women who were taking aspirin seem to have a reduced risk of getting breast cancer, but we don't know who and what, and we don't know whether it has an impact. This, by the way, is funded with your federal dollars. This came from a $10 million grant from the Department of Defense, okay, to help support this, because obviously this is a very cheap drug. It is no longer on patent, and it really does have some potential powerful opportunities, but it remains to be proven whether or not it's working. Well, what about the T cells? Okay, what about targeting the immune response to your cancer? Okay, this is a picture from a scanning electron micrograph that we had from, I've probably had this for 10 years. These are activated CD8 positive T cells, as we call them. These are T cells that are killing a melanoma target. As you can see in yellow, the T cells are in white, and you see the little kiss of death that they're providing as they're poking holes in the membrane of that tumor cell. And it takes several of them to do that and to actually kill a tumor cell. So how do you get that reaction to work? How do you harness your immune system to kill your tumor cell? So what we know, and like I said, the tumor immunity is a balancing act. This is a picture of a cancer from a patient who was on one of my breast cancer vaccine trials from a number of years ago. She's a long-term survivor from that study. And what you see in her breast cancer is these tiny little blue cells that are infiltrating into her tumor, okay, into her breast cancer, that are sort of there and poised, trying to kill the cancer, but haven't actually managed to do it. So why is that? Why does the cancer stop them? It's almost as if the immune system gets to the gates and then can't seem to actually enter. And how does that happen? How does your immune system, how does your cancer turn off immunity, and how can we block that? So there are a host of molecules involved on this, and I'm not going to quiz you on this. Some of them activate the immune system, some of them inhibit. Many of these have been targeted by various pharmaceutical companies. And what we do know, and what we've learned really much more extensively in the last 2 years, has been which cancers are now responding to immune therapy. And what I predict is that for many of you, your cancer is up on this list. And if it's not, it's probably because I've forgotten to put it up there. All right, almost every solid tumor and heme malignancy, when we look at this, there is evidence of response, okay, to immune therapies. And I think that it has surprised the medical field as to how important this has been for cancer therapy. And I do predict that there will be an integration of immune therapy as part of our overall armamentarium of surgery and radiation and hormonal therapy and chemotherapies and others, as we start to integrate immune therapy as part of our standard practice for oncology. What we do know is that many of this work was pioneered within melanoma, and then subsequently has been, many of the therapies have been demonstrated to work in lung cancer. And then now it is many of these have been migrating into other cancer types, head and neck cancer and a number of others. So as a quick reminder, using a lecture from one of my introductory immunology lectures, you have different cells that the immune system uses. When you talk about cellular immune therapy, you have B cells that make antibodies. And they're called B cells because chickens have an organ called the bursa, and that's where they were identified. We don't have a bursa, we still call them B cells. That's where it came from. And then we have T cells, and they're called that because we have this organ called the thymus. That's the little education process for the T cells. And so that's how it originally got those names. But B cells and antibodies target things outside of the cells, and T cells target things within the cell. And that's really the division of labor of the immune system. And both of these are actively involved in cancer immunotherapies. So the primary immune therapy we've had really was originally identified back in the mid-70s are monoclonal antibodies. But antibodies, okay, can target the proteins on the cell surface. And I am willing to predict how many of you have had a monoclonal antibody as part of your therapy for cancer? All right, how many? It is really amazing. And I also predict, if I am invited to give this lecture in another 2 years, that we will probably double that at least, because there are a tremendous number of monoclonal antibodies. In breast cancer, that's Herceptin or trastuzumab. In lymphoma, that's Rituxan or rituximab. But there are a host of others that are used. And what an antibody is, it's a naturally occurring thing in how we fight infections. It's what vaccines use, okay, to prevent infections. Okay, and what these monoclonal antibodies do is they're manufactured and they will target proteins on the surface of a tumor cell. And you have to identify those proteins. It's easy enough to make those antibodies, okay? And what they do is they recruit the immune system to then clear out the tumor, to kill and to clear out the tumor is part of definitely how they work. And it's a very specific interaction. So they don't stick to anything else, they just stick to the tumor. And so, but what is also evolving forward from this is something we call antibody-drug conjugates. And what that means is, how do you take an antibody and essentially add on a toxin, add on a very specific toxin. So the antibody will find the tumor, and then the toxin is there focused at the site of the tumor. And so antibody-drug conjugates are sort of the next generation antibodies that we use. We already use this in breast cancer in a drug called Kadcyla. There is a host of others that are in use and also in development. But you have this linker and then you have this toxin, and all it does is it takes what we already have that are specific for the tumor and just makes it more toxic. And we use these now, and I also predict that we will have many more of these in clinical use over the next several years. And that is and that's sort of half of the immune therapies that we think about. So what about T cells? What are T cells and how do they work, and why has it taken us so long to get there? So what T cells do is that they see your own host cells. So my PhD advisor, when I first walked into his office, he said, in his clipped, fast British accent, he said, well, we study how the difference between self and non-self. The immune system can tell, is it you or is it you that's infected? Okay, that's what the immune system, what T cells see. Has this cell been infected with a virus, or is this cell healthy? And it tries to distinguish between that very subtle difference. And so it's also seeing, is this a tumor, which is an altered self, right? It's you that's altered, versus your healthy cells, and how can it distinguish that on a molecular level? He then went on and talked about a bunch of other things. I had no idea what he was talking about. It took me 5 years to help translate it. Okay, but what you're seeing are these HLA molecules. These molecules, they are the most polymorphic region in our human genome. And these are the molecules that tell you, if you say that a kidney donor is a match or a bone marrow donor is a match, these are the molecules that say that you're a match, okay? Because they're very different across our population. It's how we as a population survive the plague and major infections, okay? That some of us will survive because we're so variable between us at our HLA molecules. And so this is how T cells see, which is why many of the T cell therapies are very specific. We may only be able to use them if you hear something like HLA-A2, it's because it's only half of us are A2, so they're only going to work for those people. So all of the T cell therapies that we develop have to be very specific for you, for your HLA, in the exact same way that we match for transplants. And so what it sees is it sees tumors. This is the actual pictures of the molecules, and I put them up there because they're beautiful. These crystal structures were solved in the mid 80s, and it transformed immunology, and they were solved literally the year that I started working in the field, and I said I could spend the next 30 years working on it. Turns out I did. And it's about finding those peptides and those targets from the tumors within them. So what is checkpoint blockade? This is a term you will hear from your oncologist. This is a term you will hear in the media. If you haven't, you will hear more of it. Okay, what checkpoint blockade is, it's how those T cells get turned off. So rampant immunity and rampant T cell immunity will be too active, okay? Your immune system, if it's seeing a virus or it's seeing something, if it gets too active, you'll have autoimmunity. You'll have cytokine release, you'll have high fevers, you'll have inflammation, you'll have fluid retention, you'll have all of these problems when the immune system gets too activated. So your immune system has this beautiful system of turning itself off if it gets too activated, and that is really the checkpoints of the immune system. And so the T cells are naturally turned off, and the tumors have figured this out. They said, well, we can just turn off those T cells using the system they already use in molecules, and there's two molecules you'll see. One is CTLA-4 and one is PD-1, PD-L1. So the PD-1, PD-L1 molecules are the ones that are targeted primarily now for checkpoint blockade for a number of different cancers. And all the blockade is, it's antibodies like we talked about that inhibit the inhibitor. That's one of the challenges, okay? It's inhibiting the inhibition of the immune system, so all it does is activate T cells. It does it non-specifically. It's going to activate all of our T cells. But because it's the tumor is expressing these molecules, it's turning off the T cells right at the tumor, we can actually deliver these drugs fairly safely, okay, with minimal autoimmunity, and to activate T cells that are specific for those tumors. And so there's been a host of immune therapies that are now FDA approved, okay, that are checkpoint blockade that activate T cells. The first one was Yervoy or ipilimumab that was approved for malignant melanoma. It hit a pathway that caused a little bit too much autoimmunity. We still use this, okay, for melanoma and for other therapies. It works in a slightly different way. Then the next one between pembrolizumab and Nivo, okay, developed by different pharmaceutical agents, okay, for melanoma, now approved for lung cancer, now approved for head and neck cancer, has been in studies in a host of other therapies, bladder cancers, breast cancers, colorectal cancers, you name it, all right? And really what they do is they activate memory T cells that are just waiting, they're sitting there at the tumor waiting to try to see the tumor, but they can't because the cancer is cloaked. It has this cloaking device of turning those T cells off. And so there are a host of new targets that are coming down the pike. But what we've learned from cancer immune therapies are, this is an example from one of the original clinical trials, is that some people respond and some don't. And those responses take a long time. And sometimes, as the immune system, as vaccines do, they take a long time to work. And sometimes we see the effects quite later. And so we've been trying to figure out as a field, how do we actually observe these trials? How do we choose these trials? How do we choose the right patients who are going to respond to these drugs? And that is where we are at right now. How do we identify who's going to respond and who's not? So in the last couple of minutes, I want to talk about something called CAR T cells, which you may have also heard about. So a CAR is a chimeric antigen receptor. What that is it's a T cell receptor that is specific for a tumor. And what they do is they take your own T cells, essentially, and make them specific for your cancer. They put genes into they take your T cells out, expand them up in culture, and try to make them specific for your cancer. And the chimeric part is this is the gene therapy part, is they actually try to fix it. And so maybe they make it look like an antibody, maybe they make it look like a T cell receptor. There are different versions of these, okay? But then they take your T cells and put them back in and have them home directly to the tumor and try to kill the tumor. They're basically taking what your immune system is already doing, souping it up and making it much more specific. The use of CAR T cells is still new, okay? It has been shown to have some really dramatic responses, in particular in hematologic malignancies such as leukemias, but we're now starting, this is how they take them out and expand them and put them back in. But now this was in a report just last month in the New England Journal on a patient with glioblastoma, okay? A cancer that's been notoriously difficult to target, okay? Responding to a CAR T cell. And so these sorts of cellular therapies are really coming down the pike, and this is one way, okay, to target the immune system for cancer, and you will hear more about these. But what about vaccines? And when I talk about vaccines, I talk about therapies, okay? Can vaccines help prevent recurrence rates? These are being studied in a number of different cancers. This is breast cancer, for example, okay? These are just some of the trials ongoing in breast cancer. But there are a number of different studies, both here at Mayo Clinic and elsewhere, on vaccines. This was from one of my vaccine trials for breast cancer. You can get inflammation targeting the tumor. You can get T cells into the tumor. You can start to get these to work. So what we're learning from these is several things. One is the immune system is incredibly powerful. It can naturally react to cancer. You can use antibodies and T cell therapies, and you can target the immune system to the cancer. You can develop vaccines to help prevent recurrence that are now in clinical trials around a number of different cancer types. But the challenge is, if you're going to harness this, and there are a host of other molecules that are coming down the pike, how do you harness it effectively and specifically when minimizing therapies? And one thing that I want to say, I like this because it just makes me smile, but one thing, if indulge me when I realized last night that I have been working on this in 30 years, this field has really undergone a transformation. And it has done this really for 15 years. What we found was it was so difficult to figure out how to do this correctly, how to do this safely, how to do it efficaciously. And it would not have gotten where it is without two groups of people, and I want to point this out. The first group has been our patient advocates, the people who have gotten there and have been telling us for 30 years that the immune system can fight cancer. And at a time when we as a field doubted ourselves, and you really helped prop that up. And I really want to say that you as an advocacy organizations and you as patients have been telling us something that we needed to hear. And you have helped keep this field forward. And the other group of people that I really want to honor is the people who have gone on these clinical trials to help figure this out. Without them, this work would never have gotten to where it is. They are our predecessors. They are the people that have led the field and have led this going forward. And so with that, I want to thank my colleagues in the Breast Cancer Interest Group. This is a collaborative effort between Mayo Clinic and Arizona State University. I want to say that Mayo Clinic has led the charge in terms of immunotherapies for decades and has really transforming this field. And I think that you are going to see some extraordinary things coming out. Thank you.